So, of the 5 stages of cholesterol biosynthesis, all the 4 stages are happening within the cytoplasm, after which the lanosterol will be formed. At the end of the stage 4, from squalene through squalene to 3 epoxide formation with epoxidase enzyme or monooxidase enzyme, squalene is undergoing conversion to give squalene 2,3 epoxide. And this squalene 2,3 epoxide then undergoes cyclization to give rise to lanosterol in presence of molecular oxygen and one molecule of NADPH. Right. So once the lanosterol is formed, it now slowly moves into the endoplasmic reticulum or microsomes, where the microsomal enzyme converts the lanosterol to cholesterol through a series of steps. Several reactions happens to finally convert the 30 carbon lanosterol to finally the 27 carbon cholesterol. So how is it happening? Yeah, squalene is first oxidized to squalene epoxide. It is undergoing cyclization to give lanosterol. It is finally converted into cholesterol. See, so this is squalene, right? So two molecules of FPP joined together. See, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. So this is one molecule of FPP. So two molecules of FPPs with the removal of one molecule of inorganic pyrophosphate give rise to squalene in presence of squalene synthase enzyme and NADPH molecule in presence of NADPH. So now after this, what happens? Squalene is converted to lanosterol with the help of the enzyme monooxidase followed by cyclase to first give rise to squalene 2,3 epoxide. So this is 2,3 epoxide, right? So you are not supposed to draw the structure after this stage, or once squalene is formed, you are not supposed to draw the structures. Up to this, you are having the structures. After the step, once squalene is formed, you just simply write the remaining reactions. You can simply write the compounds, name of the compound only. So squalene will be after only at the last step you are supposed to write the or draw the structure of cholesterol. So from this step onwards, you simply write down the name of the compound. You need not have to draw the structures, right? So it is the 30 carbon squalene is formed. This squalene undergoes oxidation to give rise to squalene epoxide first, squalene 2,3 epoxide, which now undergoes cyclization. So this is a basic difference between squalene and lanosterol. See, squalene was a linear molecule. It is not having any cyclic ring system in its structure. It's having 6 double bonds and a linear structure, right? So you can see, you can just place this FPP 15 carbon to this place. So it's simply forming a 30 carbon linear molecule, right? And after this, what happens once the cyclization happens? See, it is a crude form of, or it is almost the structure of cholesterol, but it's not exactly the structure of cholesterol, because of course the third position OH is occupied, the side chains are all there, right? But the structure is incomplete. You have to shift. See, the double bond should be in between fifth and sixth position. It's now in between the eighth and the ninth position. And it's actually a 30 carbon compound. You have to remove the 30 carbon and you need only 27 carbon atoms. So there are many additional, the two CH3 groups are there. These are the two CH3 groups. All of this have to be removed. This three there are the 30 out of the 30 carbon atoms, the three the 3 carbon atoms will be removed in the form of methyl groups. So once 30 minus 3, we get 27. So what is happening during the conversion of lanosterol to cholesterol? The shifting of the double bond happens. That is one of the reactions. The shifting of the double bond from 8th to 9th, C8 and C9 to 5th and 6th position. That's one of the changes happening. The another change is the reduction of the number of carbon atoms. The 30 carbon should be reduced to 27 carbon. So it requires the removal of methyl groups. 3 methyl groups are removed. So this is in the first step, lanosterol is undergoing demethylation reaction to give rise to 14 desmethyl lanosterol. So 14 desmethyl lanosterol. That is the methyl group from the 14th position is removed first, right? Then there is so the remaining compound, that is in order to indicate that the methyl group on the 14th position was removed. So this is the 14th position. So we were having a methyl group, then there are other two additional methyl groups here. So all these three methyl groups, this fifth carbon, 1, 2 methyl groups are there, and on the 14th carbon there is another methyl group. So the first methyl group to be removed is from the 14th carbon atom. So it's a demethylase enzyme. So in order to indicate that the 14th methyl group is removed, we name the compound as 14 desmethyl or demethyl. Methylation is demethylation is happening. So that's why the compound is named as 14 desmethyl lanosterol. See, now we are not having any methyl group on the 14th position. So the compound is named as 14 desmethyl lanosterol. Now this is followed by the removal of the remaining two methyl groups, which were on the 5th and the 5th carbon atom. So again, the two methyl groups are again removed by the demethylase enzyme to give rise to a compound known as the zymosterol. The zymosterol, right? Now, this zymo, it's again having almost the same structure. See, we are having an OH group. Then there are two methyl group side chains. They are all on the exact positions. And we are having the isoprenoid side chain also. All of it's having a double bond there. So, all of them are having the this double bond has to be removed. That will be removed only in the final step. The last step is the removal of this double bond. See, it's having a double bond here. 24 and 23rd and the 24th position. 25, 26, 24th and the 25th carbon atom. That's a double bond. This is the 24th carbon atom. This is the 25th carbon atom. So there is a double bond in between the 23rd and the 24th and the 25th carbon atom. This also will be removed, but only at the last step. So the next reaction to be happens is the removal of this double bond or shifting of the double bond from this position to this position. Okay, so that we are going to do. Zymosterol is acted upon by zymosterol undergoes shifting of the double bond from this position to this position. So this was in between the 8th and the 9th carbon atom. So it is getting shifted to the C9, C8 and C7, right? So that's known as delta 7 24 cholesterol. So that is delta 7, that is the double bond is on the 7th position and the other double bond is on the 24th position. So it is in order to indicate the position of the double bonds, this compound is named as delta 7, 24 cholesterol. Diene means there are two double bonds. So diene all OH on the 3rd position. Right, so delta 724 cholesterol. Now what is happening? Again, it is jumping from this position to this position. So it is known as desmosterol, right? So lanosterol is converted to 14 desmethyl lanosterol with the removal of methyl group from the 14th position. In the next step, the remaining two methyl groups from the 5th carbon atom is removed at a time. Say the two methyl groups are removed in a single step to give rise to zymosterol. Zymosterol then undergoes the shifting of the double bonds from the C5 from the C8 and C9 to C7 to C8 position. So the resulting compound is known as delta 724 cholesterol. It is not cholesterol, it is cholesterol. Now what happens? The C7 shifts the double bond from C7 to C5. So it is known as desmosterol. Now what is happening? What is there remaining? We have got one more double bond on the C24. It also should be removed. That is, it is to be reduced, right? So that is desmosterol is undergoing reduction to give rise to cholesterol, right? So the last double bond is also removed. So it is carried out by cholesterol. It is carried out by reductase enzyme, delta 24 reductase enzyme, right? So all these reactions are happening in presence of NADPH, right? This require the conversion of delta 7 cholesterol delta 724 cholesterol to desmosterol is resulting as a result of in presence of two molecules of NADPH and a molecular oxygen. Similarly, the conversion of zymosterol to cholesterol the reaction involves only the shifting of the double bond. So it's happening in process of isomerase enzyme, right? Now conversion of 14 desmosterol 14 desmethyl lanosterol to zymosterol. It is happening in presence of NAD or NADPH, right? Because demethylation is happening. So there are a removal of carbon dioxide is also taking place. The methyl groups are removed in the form of carbon dioxide and all. So there are the reactions and cofactors are required, and this finally it is resulting in the formation of the 27 carbon containing cholesterol. So the last enzyme is delta 24 reductase enzyme. Now, what's the what's the peculiarity of this desmosterol? This compound is highly important in nature because we have told that. The adult brain cannot synthesize cholesterol, while the fetal brain can synthesize cholesterol. So the reason is that desmosterol is present in fetal brain. So if desmosterol is present as such, it can be directly converted to cholesterol by means of delta 24 reductase. While in case of adult brain, this desmosterol is absent. That is why adult brain cannot synthesize cholesterol. While Pakshe, there is a condition where the adult brain starts synthesizing cholesterol from desmosterol, and that happens only in tumors, brain tumor. Clear? So it happens only in the patients with brain tumors that the adult brain starts synthesizing cholesterol, because only in human adult brain having brain tumors the desmosterol will be present. So that's a peculiarity of desmosterol. That's the importance or clinical significance of desmosterol. If an adult person starts synthesizing cholesterol in his brain, it is indicative of brain tumors. Okay, so actually the conversion of lanosterol to cholesterol can happen through two pathways. There are two routes through which the lanosterol can be converted to cholesterol. The first one is lanosterol can be converted to 14 desmethyl lanosterol, from which zymosterol is formed. Zymosterol is converted to delta 724 cholesterol from where desmosterol is formed. Desmosterol is finally converted to cholesterol by delta 24 reductase enzyme. Another method is lanosterol can be converted into first 24-25 dihydrolanosterol. That is, for instead of undergoing demethylation reaction first, this reduction reaction can be happening in first step. That is, lanosterol can be undergoing reduction to give rise to the 24-25 dihydrolanosterol. That is, this double bond is removed first, so that the resulting compound is 24-25 dihydrolanosterol, and after which alpha methyl delta 8 cholesterol can be formed. That is, this one, this double bond will be shifted after, then from the 8th position to 7th position, so that alpha methyl delta 7 cholesterol is formed. Delta 7 cholesterol can be converted to 7 dehydrocholesterol, right? 7 dehydrocholesterol can be finally converted into cholesterol. So the thing is that basic difference is that the order of the reaction will be changed. So there are two pathways through which the cholesterol can be synthesized from lanosterol. Either the demethylation reaction can happen first, or the reduction reaction can be initiated first. Okay, so that is how the cholesterol is synthesized in the body, on various tissues of the body. So it is almost cytosolic in nature, but the last stage, that is the conversion of lanosterol to cholesterol, happens only in the microsome, or the enzymes and the coenzymes present for carrying out the lanosterol to cholesterol formation. That is, delta 24 reductase is a microsomal enzyme. So this is present within the endoplasmic reticulum. So the conversion of lanosterol to cholesterol will be happening in the endoplasmic reticulum. That is why we have said that the biosynthesis of cholesterol happens in almost all the nucleated cells of the body. We haven't mentioned that it's happening in all the cells of the body. It's happening only in the nucleated cells, because almost all the 75% of the reactions are happening in the cytoplasm. But the last stage is happening within the endoplasmic reticulum. So for this last stage to take place, what is to be done? Only the nucleated cells will be having what? Endoplasmic reticulum. So that is why it has been told that the de novo pathway of cholesterol can happen in only the nucleated cells of the body. That too, it cannot happen in adult brain. Even though adult brain is having nucleated cells, the human adult brain cannot synthesize cholesterol under normal conditions. Only the fetal brain can synthesize cholesterol. Adult brain will be synthesizing cholesterol, but in case of brain tumors. So that is the synthesis of cholesterol. Now we'll move on to the regulation. How is cholesterol regulation happening?